Chapter 2, Section 5, Enzymes. Key concept. Enzymes are catalysts for chemical reactions in living things. Main ideas. A catalyst lowers activation energy, and enzymes allow chemical reactions to occur under tightly controlled conditions. First main idea. A catalyst lowers activation energy. Remember what you learned about activation energy in the previous section. Activation energy for a chemical reaction is like the energy that is needed to push a rock up a hill. When enough energy is added to get the rock to the top of the hill, the rock can roll down the other side by itself. Activation energy gives a similar push to a chemical reaction. Once a chemical reaction starts, it can continue all by itself, and it will go for a certain time at a certain rate. Often, the activation energy for a chemical reaction comes from an increase in temperature, but even after a chemical reaction starts, it may happen very slowly. The reactants may not interact enough, or they may not be at a high enough concentration to quickly form the products for the reaction. However, both the activation energy and rate of a chemical reaction can be changed by a chemical catalyst like the one referenced on the screen here. A catalyst is a substance that decreases the activation energy needed to start a chemical reaction, and, as a result, also increases the rate of that chemical reaction. Compare the activation energies and the reaction rates in the graph on the screen here. Under normal conditions, the reaction requires a certain amount of activation energy, and it occurs at a certain rate. When a catalyst is present, less energy is needed and the products form faster. Although catalysts take part in a chemical reaction, catalysts are not considered to be either reactants or products because catalysts are not changed or used up during a reaction. Second main idea. Enzymes allow chemical reactions to occur under tightly controlled conditions. Chemical reactions in organisms have to take place at an organism's body temperature. Often, reactants are found in low concentrations. Because the reactions must take place very quickly, they usually need a catalyst. Enzymes are catalysts for chemical reactions in living things. Enzymes, like other catalysts, lower the activation energy and increase the rate of chemical reactions. In reactions that are reversible, such as the carbon dioxide and carbonic acid reaction described in the previous section, enzymes do not affect chemical equilibrium. This means that the enzymes do not change the direction of a reaction. They just change the amount of time needed for the equilibrium to be reached. Enzymes are involved in almost every process in organisms. From breaking down food to building proteins, enzymes are needed. For example, amylase is an enzyme in saliva that breaks down starch into simple sugars. This reaction occurs up to 1,000,000 times faster with amylase than without it. Enzymes are also an important part of your immune system, as is referenced on the screen here. Almost all enzymes are proteins. These enzymes, like other proteins, are long chains of amino acids. Each enzyme also depends on its structure to function properly. Conditions such as temperature and pH can affect the shape and function or activity of an enzyme. Enzymes work best in a small temperature range around the organism's normal body temperature. At only slightly higher temperatures, the hydrogen bonds in an enzyme may begin to break apart. The enzyme's structure changes and loses its ability to function. This is one reason why a high fever is so dangerous to a person. A change in pH can also affect the hydrogen bonds in enzymes. Many enzymes in humans work best at nearly neutral pHs that are maintained within cells of the human body. Enzyme structure is important because each enzyme's shape allows only certain reactants to bind to that enzyme. The specific reactants that an enzyme acts on are called substrates. For example, amylase only breaks down starch. Therefore, starch is the substrate for amylase. Substrates temporarily bind to enzymes at specific places called active sites. In the same way that a key fits into a lock, substrates exactly fit the active sites of enzymes. That is why, if an enzyme's structure changes, it may not work at all. This idea of enzyme function, which is called the lock and key model, is shown on the screen here. The lock and key model helps explain how enzymes work. First, enzymes bring substrate molecules close together. Because of the low concentration of reactants in cells, many reactions would be unlikely to take place without enzymes bringing substrates together. Second, enzymes decrease the activation energy. When substrates bind to the enzyme at the enzyme's active site, the bonds inside these molecules become strained. If bonds become strained or stretch slightly out of their normal positions, they become weaker. Less activation energy is needed for those slightly weakened bonds to be broken. The lock and key model is a good starting point for understanding enzyme function. However, scientists have found that the structures of enzymes are not fixed in place. Instead, enzymes actually bend slightly when they are bound to their substrates. In terms of a lock and key, it is as if the lock bends around the key to make the key fit better. The bending of the enzyme is one way in which bonds in substrates are weakened. Chapter 2, Section 5, Enzymes. Key concept. Enzymes are catalysts for chemical reactions in living things. Main ideas. A catalyst lowers activation energy, and enzymes allow chemical reactions to occur under tightly controlled conditions.